Hello, everybody. Standing by for today's briefing from Geneva WHO on the composition recommendations for the vaccine, influenza vaccine for the season 2023-24, and avian and zoonotic influenza. Give us just 2 more minutes to finally set up the system here, and then we're with you. One. Hello and welcome, finally, to today's virtual press conference here out of WHO headquarters in Geneva. We have 2 parts to this press briefing today, and let me run you through this. We have, first of all, the WHO recommendation on the influenza vaccine composition for the Northern Hemisphere 2023-2024. And in a second part, we'll discuss the zoonotic avian influenza situation. And since we had updates coming last night in the media, this might be a very interesting part. Like all other parts will be interesting. So thank you for standing by for this. Let me introduce the panel, and we have a strong panel here today. First and foremost, we have Dr. Sylvie Briand. She's Director for WHO's Epidemic and Pandemic Preparedness and Prevention. Then we have Dr. Wenjing Zhang. She's the head of the WHO Global Influenza Program. We also have Dr. Kanta Subaru. She's director of the WHO Collaborating Center for Reference and Research on Influenza at the Victoria Infectious Disease Reference Laboratory in Australia, and she will walk us through the main recommendations on the influenza vaccine for the next winter season in the Northern Hemisphere. We also have Dr. David Wentworth. He's director at the WHO Collaborating Center for Surveillance, Epidemiology and Control of Influenza at the CDC in the United States. To my left side, we have Dr. Richard Webby. He's director for the WHO Collaborating Center for Studies on the Ecology of Influenza in Animals, Department of Infectious Diseases at St. Jude's in Memphis in the United States. And last but not least, on his left side is Dr. Francesco Bonfante, an expert on zoonotic and avian influenza at FAO and with OFLU. With this, everybody, thank you very much. We're recording this and we will afterwards make the recording available. You can also watch us as you may be doing live on social media. So welcome everybody. Enough from me. And I hand over to Dr. Briand for the opening remarks.
Thank you, Christian. Good morning, good afternoon, and good evening. Before we get to the influenza vaccine composition, I would like to briefly address the reports of human H5N1 infections in Cambodia. Cambodian authorities have informed us of 2 confirmed cases of avian influenza H5N1, both members of the same family. One of the cases, an 11-year-old girl, unfortunately passed away. We are in close communication with the Cambodian authorities to understand more about the outbreak. Field investigations are ongoing. The global H5N1 situation is worrying, given the widespread of the virus in birds around the world and the increasing reports of cases in mammals, including humans. H5N1 influenza is a severe respiratory disease with a range of symptoms observed from mild to fatal. The mortality rate among cases reported with H5N1 infection over the years is over 50%. WHO takes the risk from this virus seriously and urges heightened vigilance from all countries. Now on the vaccine composition. This week, WHO held an important meeting in Geneva and the influenza vaccine composition meeting. Influenza viruses are constantly evolving, and there is a risk that they escape the immunity conferred by the vaccine. Therefore, we need to update the vaccine twice a year to make sure that we have the best possible vaccine to protect the populations around the world. During this meeting, top-notch experts from WHO collaborating centers, national influenza centers, regulatory laboratories, and also laboratories from the animal sectors reviewed the circulating influenza viruses, assessed the change compared to the last influenza season, and advised about the vaccine composition. They are looking at viruses circulating in human population, but also at viruses circulating in animals. And I would like to thank the colleagues from FAO, OHA, and OFLU for their critical contribution to this vaccine update. This morning, we also had an information session with vaccine manufacturers to share the recommendation of the experts so that they can immediately start the production of the vaccine for the Northern Hemisphere season. The vaccine composition meetings makes recommendation on the vaccine virus for seasonal influenza epidemics, but also for the candidate vaccine viruses for zoonotic influenza. The GISRES, the Global Influenza Surveillance and Response System, is a global network of laboratories and partners working year-round to monitor the influenza situation and the related viruses. And based on this global collaboration of scientists and institutions around the world, we are able to assess the risk of emergence of novel influenza viruses and prevent epidemics and pandemics by implementing early containment measures in the case of zoonotic virus become fit for human-to-human transmission. But based on the recent reporting, we are reviewing the risk assessment. Thank you very much. Back to you, Christian.
Thank you very much, Dr. Briand. We'll give right to Dr. Wenjing Zhang, the head of the Global Influenza Program at WHO.
Thank you very much, Christian. So to address the pressing public health issues highlighted by Dr. Briand, they need a global system. And that system is called the WHO Global Influenza Surveillance and Response System, or GISRES, currently comprising 159 institutions in 127 countries, areas, and territories. While you just can see us a few presenting in this media briefing, but behind us, there's a global system of people from developing and developed world working year-round and in several generations in the past 70 years. This is the real and true capacity or strength that the global system, the GISRS, has. With the support from member states, with the global agency, the GISRS is being developed into a stronger system, GISRS Plus, so that GISRS would be able to address not only the influenza, but also other emerging respiratory threats or epidemic and pandemic potential. So let me just stop here.
Thank you very much. And we move to Dr. Kanta Subarao, and she will walk us through the recommendations.
Sure. Thank you very much. As Wenqing explained, we rely very heavily on data from around the world, from the Global Influenza Surveillance and Response System, which, as she's explained, is a tiered system with national influenza centers, the WHO collaborating centers, and the WHO essential regulatory laboratories, as well as some H5 reference laboratories. And as we've indicated, in the case of the vaccines for zoonotic or pandemic preparedness purposes, we rely heavily on interactions with our colleagues from the animal sector. But I'm going to talk about what we do for the seasonal influenza vaccine composition. And we meet twice a year, once in February, to discuss and make recommendations on vaccine strains to be included in the Northern Hemisphere vaccine for the following upcoming winter. And that's what we've done this week. So we spent 4 days looking over data generated from around the world, shared with the WHO collaborating centers and essential regulatory laboratories, where we examine the disease activity in different parts of the world. We look at virologic surveillance, where we characterize viruses for their genetic characteristics and antigenic characteristics, where we, as Wenqing said, we're looking for evidence of new variants that might escape the immunity conferred by the previous year's vaccine. And that is critical in determining whether we should update vaccine candidates. In addition, we have panels of sera that we share and exchange between the centers, where we look to see whether people that got the previous year's vaccine have antibodies that would cross-react with newly emerging clades of viruses. So the last piece is that we also look at antiviral sensitivity of viruses that have been circulating around the world. So based on all of this, we look at the influenza A H1N1 PDM09 viruses, influenza A H3N2 viruses, and the two strains of influenza B, two lineages of influenza B viruses, the B Victoria lineage and the B Yamagata lineage. We have not seen circulation of influenza B Yamagata lineage viruses, and we have made recommendations at the conclusion of these discussions to update one component, and that is the H1N1 PDM09 component. Thanks very much.
Thank you very much. And of course, we will send the list of recommendation right after this briefing. So for all those who couldn't keep track now, but thank you very much. Now, as we said, we have a second part to this. Also, we'll talk about zoonotic and avian influenza today, and to update you what's going on what's happening around zoonotic influenza in the frame of pandemic preparedness, we'll have Dr. Richard Webby to give us the latest.
Thank you, Christian, and hello all. So, yeah, as we've just heard, an integral part of what's gone on in the past week is looking at influenza virus activity in the animal populations of the world within the context of pandemic preparedness, and particularly within that candidate vaccine virus production. So, as some of you may know, the World Health Organization maintains a list of candidate vaccine viruses, or seed viruses if you wish, that covers the diversity of influenza viruses that are considered risk for public health. And so we sit down in this meeting with many of the same techniques that you just heard about from Dr. Sobrao, and look at, one, what we have already in terms of candidate vaccine viruses against risk viruses circulating in animals, and whether the evolution of viruses in those animals has come to a point where we need to recommend new strains to cover some of that diversity. And so that happened. To sort of run down some of the numbers from this in terms of infections, and this is since September, I would categorize these numbers as fairly typical for over the past few years, despite a huge amount, an increase in particularly H5 activity in bird populations around the world. So this particular period, there was 9 H5 cases reported. So that brings the total since 2003 to 965 cases of H5 infection. There were 8 H9 and 2 virus infections reported, and 7 viruses, what we call variant viruses. So 7 viruses coming from swine populations into humans. And so we looked at these, again, infections, zoonotic infections, in the context of what we know is circulating in animals. We did make one recommendation to update the available candidate vaccine viruses for our H5s, and this is really in response to the spread of H5, particularly through the Americas, and a little bit of evolution of viruses associated with that spread. So we are putting another H5 candidate vaccine virus into production, and that will start soon. So, I think that's probably a good place to stop.
Thank you very much, Dr. Webby. And now let me ask Dr. Wentworth or Dr. Bonfante if there's anything to add. Dr. Wentworth?
I actually don't. I think that the description of what we did with the seasonal was right on, and I don't really have much to add. I guess one other part that I want people to understand is we also do a lot of heavy data integration of all of those components for both the human seasonal viruses and zoonotic viruses, and this includes fitness forecasting. So not only what's happening now, but what things are predicted to move forward into the future. And of course, with that kind of an approach, the more near time forecasts are always more accurate than far out into the future.
Thank you very much, Dr. Bonfante.
Yeah, I think Dr. Webby mentioned all of the crucial aspects that we touched upon in the last 5 days.
Thank you. With this, we can now open the floor to questions. And as usual with our webinars, please use the raise your hand icon on your screen in order to get into the queue. Happy to see old familiar faces, so to say, on the screen here. And Helen Branswell from Stat News. Helen, please go ahead.
Hi, you unmuted Aaron Prater. Is that okay if I ask a question? Hello?
Hi, Christian, I'm also unmuted. How do you want to handle this? Christian, hi, can you hear me?
Yes, now we have you.
Okay, thank you very much. At the beginning of the session, Dr. Briand talked about the fact that there have been 2 cases confirmed in Cambodia. There were reports out of Cambodia yesterday that they had identified a number of contacts, I believe a dozen, and were testing them. Can you tell us if any of those people have been eliminated as cases, or is it just that the testing results are not yet in for the other 11 people? Thank you.
Thank you very much. Do we have Dr. Briand, Dr. Webby?
Well, thank you for this question, Ellen. As we are speaking now, they are having a teleconference with Cambodia currently, and so the tests were ongoing. So we don't have yet the results. So as soon as we have more information, we will communicate about it. but 12 contacts were sampled, and so we are waiting for those results.
Thank you. Yeah, it's really brand new, everything. So next question goes to Erin Prata from Fortune. So Erin, now it's your turn.
Great. Thank you so much. Yes, this is Erin Prater from Fortune. I just wanted to clarify the teleconference right now that's happening with Cambodia. Is that with WHO officials, or is that Cambodia with other parties? But my question was going to be, I'm reading the second man who was diagnosed was this girl's father. Did he also have contact with birds? Any idea if this was transmitted to him from the girl or from birds? Thank you.
Thank you, Erin. The latest, let's see.
Okay, thank you. On your first question, this is a WHO Cambodia call based on the IHR requirement, because as you know, based on the international health regulations, we have a joint risk assessment with countries' authorities. And so this is currently what we are doing, discussing with Cambodian ministries of health to get the latest information and the risk assessment. Regarding the case in the father, so the father was a contact and had symptoms, so that's why he has been sampled. But so far it's too early to know if it's human-to-human transmission or exposure to the same environmental conditions. And so that's why we need more investigation to clarify the mode of transmission. Thank you.
Dr. Zhang, please.
It might be helpful also to note that highly pathogenic avian influenza H5 has been circulating in Southeast Asia since its re-emergence in 2002-2003. So we need to put that into context. There being viruses were detected or isolated from birds in Southeast Asia. And we also have, we've been working closely with the World Organization for Animal Health and the FAO also to monitor on the animal front. And actually we have experts here, maybe you can provide.
Yeah, as Dr. Zhang just mentioned, Southeast Asia has been facing circulation of highly pathogenic avian influenza viruses of the H5 subtype for many years now. And in this area, we know that circulate different genetic groups of viruses, and vaccination for H5N1 viruses is implemented. It's been implemented for many years now, and there are important national surveillance plans overarching across human and avian health, and providing timely surveillance in these regions. So we will have to conduct a thorough, as was just mentioned by Dr. Briand, risk assessment. And as soon as the additional information regarding the genetic type of virus that has been causing these zoonotic infections, will be in a better position to assess the risk posed by these virus. Over.
Thank you much, everyone. Before we go to the next question, let me please remind journalists who want to get into the queue, please identify yourself not only by name, but also by outlet, because it's difficult to determine who you're working for if we don't have your full details here. Next question goes to Antonio Brotto from EFE. Antonio, please go ahead and unmute yourself.
Thank you, Christian. Well, you said it, but I'm Antonio Brotto from EFE, Spanish News Agency. I don't know if this is the right meeting for my question, but I would like to know how near is the world from developing a vaccine that can protect both COVID and flu? That's my question. Thank you.
We can try if we find an answer for this one. Where do we start here? Dr. Kanta Subaro, please.
Yeah, I think this is a question that a number of manufacturers have been considering, and many public health agencies are considering as well. very often from the sort of practical logistic reasons of the ease of administration of two vaccines if they're concerned about a winter outbreak of respiratory illness. However, I think at this point, we know that they're very effective COVID-19 vaccines and influenza vaccines. There are few manufacturers who I think are looking at combinations, but I think the bulk of the programs will be administration of two different vaccines. Whether they're given around the same timeline is something that each national authority will determine. I don't know if the others want to add anything.
You want to add? No, then thank you very much. I see Helen has your hand up for a follow-up. So Helen Branswell, please go ahead and unmute.
Thank you very much. This might be a two-pronger. Is there any data yet on the genetic sequence of the virus from the girl in Cambodia? And the second question relates to the updating of the strain for H5N1 vaccine. Has any work been done to look at whether existing stocks of H5N1 vaccine would work against contemporary viruses? Thank you.
Yeah, I'll first start with Dr. Briand on the details, although I think we have too little information yet, but please go ahead and then we go to Dr. Verbier.
I think the first question was on the genetic sequence, so maybe Richard can address it.
Then perfect, Richard.
Yeah, hey Helen. So no, the information is not yet available on the specifics of the human case, but just to sort of add on top of what Francisco said, in Cambodia over recent years, they have reported in their poultry through the H5 reference lab and others in country, there are multiple clades of H5 circulating. That includes the 2344B, which is the one that's spread through Europe, into Africa and the Americas now, but they also have a 2321C, which is a lineage that's been a little bit more localized in that region of the world. So, right now we can't make the determination or even what the likely outcome of that is. In terms of your other questions, it's a great one. So, there are some stockpile vaccines, some trials that have been done, and there has been a little bit of work looking at some of those. So serum collected from people who took part in vaccine trials to some of these earlier H5 clades, and the serum for those people actually reacts quite well with some of these recently circulating viruses, which, again, from a stockpile vaccine or response point of view, I think is encouraging. That suggests that the human response to some of these vaccines, if it's an adjuvanted vaccine at least, does induce a broad immunity that cross-reacts with a lot of these clades that we're seeing. So from that perspective, I think it's encouraging.
And Dr. Wenjing Zhang.
So I would also like to add that for H5 vaccines, so far they are close to 20 such product licensed for pandemic use. And WHO, as Dr. Richard Webby just said, we've been really monitoring the evolution of the virus, conduct the risk assessment, and update the candidate vaccine virus that will be used or be updated into their product.
Yes, and there, in 2003, there were some countries who constituted stockpiles of H5N1 vaccine, because at that time, as you remember, maybe there was fear that this virus could become a pandemic virus. But since then, we had in 2009 the H1N1 pandemic, and so the risk assessment shift, of course, and vaccine production was more geared toward producing more H1N1 vaccines. So, which now this virus H1N1 vaccine is part of the seasonal vaccine. So. those stockpiled at that time were very useful because, as Richard mentioned, we were able to administrate them to certain humans and so see what was the reaction, the safety of the virus, but also the efficacy of the virus. And as he mentioned, the efficacy is still quite good with even the circulating viruses we have now. But however, we are monitoring the situation to make sure that if there is too much evolution of the virus, we can also update the vaccine. What is important is that the work that is being done by GISRES and the expert on the vaccine composition is absolutely instrumental to reduce the time lag between the discovery of a virus and the real production of the vaccine, because having a sort of a library of vaccine viruses, candidate vaccine viruses, shorten the time of production. So it allows us to be much more reactive in case of the emergence of a novel virus.
Thank you all very much. Next question goes to Alexander Tin from CBS News. Alexander, go ahead and unmute yourself.
Hi, it's Alex Tin from CBS News. Thanks for calling on me. Following on Dr. Webby's comments, can you help us understand how that squares with the 4 to 8-fold reductions to some viruses in the Americas that Dr. Davis described earlier today? And can you also clarify how changing the candidate affects the timeline of production? Thanks.
Yeah, so again, we're going to think of this in a couple of different aspects. So when we do the updates of our candidate vaccine viruses, which you referred to, Todd Davis reported on or summarized this morning, we are really more focused on the finer details of these viruses. So we use serum that's produced in ferrets, which is quite discriminatory. The idea being we want to get sort of into the weeds of how different these viruses are. But what we've seen in the context of this, but also sometimes in the context of the seasonal decisions as well, sometimes the human response can be a little more broad than that ferret response. So one is really more from a risk assessment, finer detail analysis of what's circulating. The human response, I think fortunately, it tends to be a little bit bigger, but I'll say we need a whole lot more work like this to understand exactly how broad that response is. We've also got to keep in mind the study that I talked about that came from a vaccine that was adjuvanted in humans, which again gives a little broader response. The work that Todd described this morning, typically doesn't include that type of reagent as well. I hope that answers your question. And that answers one of your questions. If yes, I'll go into the second. Yep. Okay. The second, right now I don't think it changes timelines of anything per se. So, many of these reagents that we produce, these updating of the candidate vaccine viruses, are really done for preparedness purposes. So, they're not necessarily pushed through into manufacture or even trial lot production immediately. So, the impact on timelines is in terms of real response is limited. If the question is coming from a point of, well, if viruses represented by this new CVV start to take off tomorrow, then I guess we would still be 4 or 5 weeks away from having that reagent developed. But at the same time, the ones we have on the shelf now do a pretty good job against these circulating viruses as well.
Thank you very much. Looking around for any add-ons? No. So next question will go to Claire Wilson. But Claire, please identify yourself where you're working for.
Hello, this is Claire Wilson from New Scientist magazine. Could you tell us what would be the next steps if we got the really bad news that the bird flu viruses had started being easily transmitted between humans? What would happen next? Thank you.
Yeah, big speculations. Let's start with Dr. Briand and then have others chip in here.
So, and this is why we are monitoring the situation very carefully, because there are some very short-term activities and interventions that we can put in place. first to reduce the human-to-human transmission by treating cases, isolating cases so that they don't further transmit the virus, but also identifying contacts. And we can use antivirals and to treat those contacts for post-exposure prophylaxis and reduce the likelihood of further human transmission as well. We will also, of course, try to control the source of the virus in the animal population so that we have also less risk of spillover from animals to humans. And so these activities, of course, will be focused in the areas where we see the novel virus starting to have this capacity to transmit from human to human. And there will be enhanced surveillance in this area so that we can make sure that this stays contained in this geographical area. And so while we are doing these very early containment activities, we will also obviously look at the potential extension of the transmission and get the neighboring provinces or countries ready for potential human-to-human transmission as well. And basically, we have 5 types of intervention. Of course, we will look at the surveillance and really have our surveillance protocol in place to be able to detect any signals, but also to monitor the circulation of the virus and the disease and its impact on population. We will also, secondly, have protect the communities, inform people, and make sure that they know what are the risks and what they can do about it. We will also, of course, strengthen the care system to make sure that they can attend the patient and also protect healthcare workers. And we will also start the development of countermeasure, be it antivirals or vaccines. And finally, we will ensure that there is a very good collaboration and coordination between sectors, between stakeholders also, and at national, regional, and global level. So we are not yet there, of course, but we are. That's why we are starting to exchange information so that everybody is aware of the risk and we can very quickly react and put in place those interventions.
Thank you so much. Next question, we go to Thomas Mekwan, if I got the name right. And Thomas, please unmute yourself and identify yourself. Thomas, can you hear us? You're still muted. As we can't get to Thomas, we'll move next to Sarah Taisir Bencheri from Politico. Sarah, please go ahead and unmute yourself.
Hi, good afternoon. Thank you so much for taking my question. I have 2 questions. The first is, am I understanding it correctly that the H5N1 vaccines that are approved and available on standby right now would be expected to be sufficiently effective against the strains of avian flu that are currently circulating? And if so, like, what's the primary outcome or metric? Is it that they're effective? They're expected to be effective to reduce transmission, to reduce severity of disease, to reduce mortality. So that's the first question. And then the second one is just in terms of surveillance. I mean, one of the common themes we've been hearing about in the last, I would say maybe 6 months or a year with the COVID pandemic is that the surveillance globally has been going down. And I'm just wondering, to what extent is that affecting surveillance of other pathogens like avian flu? If you could just comment on surveillance, perhaps specifically to Europe or globally. Thank you so much.
Thank you very much. Let's see if we can track all these questions. We'll start, Dr. Webby.
Right, Sarah, maybe I can have a crack at the first one. So I think we've got to be a little bit careful again in terminology here. So the studies I talked about were serum taken from people who took part in trials of these vaccines and just looking essentially in laboratory tests how well they recognize or neutralize the current batch of viruses in that assay. And that we think informs somewhat what the real world efficacy or effectiveness of that vaccine would be in humans. But making that direct one-to-one leap, I think, is a little bit dangerous at this time. I think clearly moving forward, the best matching vaccine, should something happen, is the way to go. But there is going to be a period of time when that perfectly matching vaccine won't be available. So that's when the more stockpiled vaccines perhaps could come into play, and offer certainly probably protection from the more severe disease. So, hopefully sort of the most severe outcomes. Would it stop infection? Would it stop transmission? Again, probably unlikely, but again, that's a lot of hand waving there. So just I think we need to separate in vitro reactivity of people's antibodies versus vaccine effectiveness.
Thank you, Richard.
From that part, yeah. Dave,
now, yeah, David, maybe.
Yeah, I'll just, this is Dave Wentworth, I'll comment as well. I mean, I think Richard really hit the nail on the head. And as Dr. Briand pointed out, I mean, I think one of the things that I'm hearing in throughout these questions is really the ability to go to the library of vaccine viruses that already exist, eliminate 6 weeks to 8 weeks from their creation. And so the pandemic preparedness or readiness efforts that the committee puts together on this are quite important. And so, for example, the clinical trials that Dr. Webby was pointing out were to an H5, which is in this 2344B clade that has circulated for quite a while, but it was actually from nominated as a vaccine candidate many, many years ago. And then it had to go through these clinical trials. Since then, another one was nominated. It was nominated a couple years ago. And that one is even more closely related when you use that fine-grained ferret immunology to assess. And as Dr. Webby pointed out, humans tend to have a broader response. We also tend to have a lot of immunological memory to many parts of the virus that help to protect us from disease, not just the key component of the vaccine, which is the virus attachment protein. And so I think when you started to talk about what the goals of the vaccine would be or what would be expected, a lot of it has to do with how, of the library of vaccines that we can pick from already, what could be used immediately, and then start phasing into what would be an optimal candidate as a potential outbreak unfolds. And so that may not be a particular strain from the new, the brand new one, but it might be unfolding a little bit later. And then with regard to, I think you can really just consider what we understand about immunology. We expect better protection from disease, and that protection from symptomatic disease is more difficult bar to achieve. Protection from transmission, which be a great goal, is also very difficult to achieve. And that's where that's more dependent and correlated with highly potent neutralizing antibodies that would be expressed in people that have been vaccinated, particularly if they were in areas where you inhale the vaccine. So if they are IgA type antibody induction. So it runs a range that can happen. And I think the second part of the question I've lost, but surveillance. Oh yeah, I did want to touch upon that. So there's two aspects, and Dr. Webby hit it on the head when he described how we work, particularly with the animal sector in collaboration at the animal-human interface, and then for surveillance of animal viruses. But the other component that's really true in influenza and part of the GISRIS activities that really make up the warm base of influenza that maybe is a little different than many of the other pandemic threats we face is almost all of the things we use to detect seasonal influenza will help us detect pandemic influenza. So they, when we're screening humans for seasonal influenza viruses, we a subset of those we can really guarantee will be subtyped, so we'll know if they're like normal human seasonal viruses. And if they react with two different parts of the assays, and so we can tell they're type A, but they don't look like typical human influenza viruses. And so those can be employed right away, both from a diagnostic perspective to track and survey influenza viruses in people. So it helps us more rapidly identify a transmission from an animal reservoir to a human, known as zoonotic transmission.
And Dr. Wenjing Zhang, please.
So I'll add a little bit more on your question about surveillance. Surveillance for influenza was disrupted at the very beginning stage of the COVID-19, but it returned to normal, and now is doing much better. Actually, throughout the COVID-19 pandemic, the human infection of zoonotic influenza, including avian influenza, has been detected throughout the COVID-19 pandemic, although the seasonal influenza activity was low at the very beginning of the COVID-19 pandemic. So currently, the surveillance system, they are using multiplex, i.e. that will be able to detect both influenza and SARS-CoV-2 at the same time. So they can do both. And also, I would like to add that after COVID, a lot of other systems, for example, the system of pneumonia with unknown etiology in some countries, they are actually quite engaged. So if there's any infection of the emergent, including avian influenza virus in those patients, it will be able to pick up. The last but not least point is our collaboration with OFLU, with the World Health Organization for Animal Health and FAO. We started since 2005 on the avian influenza. So this is the real ground of work. We are working continuously, day to day. So this is a surveillance. In WHO, we are really very confident with the global influenza surveillance and response system globally and with partners around the world to monitor and to surveil on the front of influenza.
And Dr. Briand?
And just to complement that, if we have the emergence of a novel influenza virus, of course, it's very important to know the virus and its capacities, but also it will be very important to have good surveillance in humans because it will be a novel pathogen and the impact it can have on human health might be different from what we know from seasonal influenza viruses. So it will be very important at the beginning to have very good clinical reports on the first few cases so that we can assess the impact on human health and the impact, also the fact that because it's a novel pathogen, we won't have as much immunity as we have for seasonal viruses. So the clinical symptoms may be very different. Thank you.
Thank you all very much for extremely interesting replies here. Next question goes to John Pinkstone. And Joe, please identify yourself and then unmute. No, first unmute and then identify. One of the two things didn't work. So thank you very much. We'll try next. Aaron Prater from Fortune has a follow-up. So Aaron, please go ahead.
Thank you so much. I did want to loop back to the girl in Ecuador who had H5N1. I think on last check she was recovering. Has she fully recovered? And just to double check, did anyone else in her area, was anyone else sickened? Also, what kind of antivirals are being used with these H5N1 patients? At least I believe that girl was on antivirals, and how effective are they? Thank you.
Yeah, let's see what details we still have, Dr. Briand.
Yes, the girl recovered and she's out of the hospital. Indeed, as you rightly mentioned, we have antivirals for treating influenza cases. So, of course, you know oseltamivir, which was used since 2003 in previous H5N1 outbreaks. Either to treat cases or as a post-exposure prophylaxis for contacts. But now, since then, we have had many new generations of antivirals, and we have antivirals for outpatients that can be administered very early on in the course of the disease, but we also have other antivirals that can be administered to more severe patients, even in IV, We have a full range of new antivirals, and we are working very closely with manufacturers of those medicines to see what kind of product they can provide, but also how fast they can scale up their production in case we need more of those drugs in emergencies.
And thank you, Dr. Briand. Now we'll try one more time for Joe Pinkstone. Joe, again, please unmute and identify yourself. So we see the microphone is open, but we don't hear you. So Joe, thank you and sorry for that. Then the last question for now goes to Ashwin Barshinge from Observer Times in India. Ashwin, please unmute yourself and go ahead.
Thank you for considering my question. My question is, what are the future challenges for development of zoonotic vaccines, and what steps WHO considering to reduce procedural time factor in order to get final vaccine candidate for prevention of zoonotic diseases, which will be endemic or pandemic? Thank you.
Let me look around. May we start with Dr. Webby?
Maybe I could tackle the challenges question. The challenge, the single biggest challenge we have to the zoonotic influenza question is the fact there is just such a huge diversity of influenza viruses that are circulating in some of the bird and other animal populations of the world. We've been talking mostly here about the H5, which is just one of the groups of viruses that circulate. And with most of our current vaccine technologies, as I'm sure you're aware, they're fairly narrow in terms of that breadth of animal viruses that they will actually protect against. So that's where a lot of research, of course, has been going on for many years, is to try and improve the way we immunize against influenza and try and induce a little bit more broad immunity than what our current approaches do. But single biggest problem is so many viruses, so many different flu viruses in those animal populations.
Let me look around. Dr. Briand? Oh no, Dr. Sabaro, please go ahead.
Yeah, I will just expand a little bit on what Richard said. So there's this great diversity of viruses in the animal populations. And so the goal, the sort of holy grail is to try to have a universal influenza vaccine that would protect against many different subtypes. There's an enormous amount of research and very exciting scientific research into the development of universal influenza vaccines, but there are many challenges in that regard. It is identifying shared or conserved targets across a range of influenza viruses of different subtypes. And once you identify what those targets might be, to develop vaccines that will elicit immunity against those in a way that can be done rapidly. So there's a lot of effort that's been going on for many years. It's been very high up on research agendas. Unfortunately, we're not yet at the point that we have something that is there yet. But I think that is where there's a tremendous amount of hope and research. Thanks.
Another challenge is also the switch in production between seasonal vaccines and potential pandemic vaccine. Because currently our experts have put forward some recommendation for the seasonal vaccine production. So manufacturer will start the production of this vaccine for the next fall vaccination campaign. And if a new virus emerge in the meantime, depending at the stage of this production process, it will be complicated maybe to switch from one type of vaccine to another type of vaccine. So this switch issue has been discussed for many years now, since 2009 actually, because we were faced with this kind of challenge as the H1N1 virus in 2009 emerged. I mean, we discovered it in April, and the vaccine production for the Northern Hemisphere, full Northern Hemisphere, was already ongoing. So very hard to stop the production at this stage. And so we had to consider when to stop it and how to do this transition.